Thank you to the organizers and the sponsors of this event. So for those of you that are not familiar with Calimmune, we've been around really since 2006. Didn't really get our full launch until 2008 from technology that came out of Dr. David Baltimore's lab, Irvin Chen's lab in Inder, Verma, recognizing that ex vivo autologous stem cell gene therapy hadn't really been born in the process that they believed needed to be in order to be a viable commercial option. Since that time, we started in HIV and ended up working with first generation technologies, and I'm going to share with you where we're headed as a company. Learning, we were the first group in the world to do outpatient stem cell gene therapy with a conditioning regimen, and there's been a lot of processes that have gone into really learning not only what's necessary to make the best cell product, but also what's going to be necessary to have this work at a patient level. So for us, when we think about next generation technology, we're not only talking about the tools and the skills necessary to get there, but what it's really going to take to get to the patients. And we'll come to that. We have tools both in manufacturing as well as the ability to make this an outpatient stem cell gene therapy process, which we are looking at our first gene therapy in sickle cell disease with this next generation technology. You'll see we looked at the field of HIV. It's a very difficult space to begin with. Our founder being in HIV with David Baltimore ended up paving the path. We learned a lot about not only these types of cells, the stem cells, but what it's going to take engraftment-wise, the variability between patient to patient to really make this work. So it's clear to say that there's been a tremendous amount of progress in stem cell gene therapy. As many of you know, Strimvillis, which was approved last year, ADA SCID, moving forward the whole space. It's the first stem cell gene therapy that's been approved. It's a very exciting time, but one of the obstacles is that that's a therapy that is an inpatient procedure today. You have to go to the location, stay there, and for patients, both their families and children, it's difficult. Good news is that it's curative. Bad news is to be able to distribute something like this worldwide, you have to have a number of gaps that you bridge. And we're looking at that as a company. One of the biggest gaps is the toxic preparative regimens, the conditioning regimens that sometimes with the adverse event profile makes it difficult for patients to even consider this as a path. These can cause adverse events like sterility and infertility, but even more so, it compromises the immune system and requires patients to be hospitalized. It's also expensive, and in an inpatient setting, not all the hospitals in the world would have the capabilities of distributing this. Not to say that there shouldn't be a distribution channel, but it just makes it much more difficult to begin to bring this to more patients in the settings that I think we all aspire. So the concept here is pretty straightforward. To go from an inpatient setting where you've got autologous stem cell gene therapy where patients are being treated not just in the hospital but have to stay there and have their immune systems really reconstitute, but going to an outpatient setting. And there's a number of things that we are essentially planning on applying. The first is, in order to bridge the gap of the time period necessary for patients to stay in the hospital and make it something like an outpatient therapy, you have to be able to increase the engraftment rate without increasing the amount of conditioning. To do that, it's a feat. And we have a new technology that aims at doing just that. It allows for cells to be given back to patients with a reduced intensity conditioning regimen and then titrate up based off of a chemoselectable technology in order to reach the expression level necessary for the genetic payload. So we call that technology Select Plus. It goes into a cassette. We are working in lentiviral stem cell gene therapy applications, and today it's in lenti that we have started this process. It allows us to actually put in the same application where we've got the payload, this unique cassette that makes the cells chemoselectable. Again, being able to give the cells back once the patients go home, be able to ultimately titrate up the amount of engraftment within the patients. This is a unique aspect to this type of technology. We have additional proprietary methods. Many of our members of our team have been working in the stem cell gene therapy space for years, and this is also an important factor, not just the quality of the product, but the collection processes, the manufacturing, and the like. And then lastly, we have a system, a stable production system, that allows us to produce lenti in bioreactor scale. And knowing that this is a space where we're looking to aim at more than just tens or hundreds of patients becomes an important and critical factor to be able to really grow into the scale necessary for manufacturing for commercial viability. So this is how the process works. For many of us here in this room, we're very familiar with autologous stem cell gene therapy, where the cells are taken out, collected either through apheresis, in some cases it's through bone marrow aspirate. The cells are then transduced, and during this time period, oftentimes the patient is hospitalized because they have to go through conditioning regimen that requires them to be in the hospital for at least 4 days. What we have done is we've been able to identify a path where there would be a reduced intensity conditioning regimen during this time period in an outpatient setting. And then the patients would come back and receive their own cells, and they would have the ability to go back home. We ended up testing an outpatient conditioning regimen in the HIV setting with 12 patients. Once they go home, after about 4 to 8 weeks, they'll recover. Their neutrophil counts will come back up based off of the reduced intensity conditioning regimen, and then they'll be able to take a pill that's actually got a well-known profile, that the toxicities are well-known and the pharmacokinetics are well-known in order to titrate that particular level of expression to the level necessary per patient. Again, patient-to-patient variability in our space, as we all know, can be a difficult obstacle. So again, we started in HIV. We're planning on now moving into sickle cell disease. And as we look at the horizon, of course, beta thalassemia, but there's other diseases where hematologically patients need to have correction at a stem-based level. So some of the benefits, just going and taking a step back, looking at the selection technology, one of the things that you ultimately look to achieve is a high level of chimerism. It's difficult at times to know before giving cells back how well the patient's niche is going to accept the cells that you give back. And in fact, oftentimes, after a number of months, what will happen is engraftment levels will stabilize and then work within a range, meaning that after 3 to 5 months, what you'll see is that they'll come down and work within a % range, a margin of being able to express the necessary gene modification. In the case of these mice, what we ended up doing is treating with a selection criteria, and we did that at different doses, but essentially we're able to reach a high level of chimerism both in the bone marrow, and then we're able to also increase this up in peripheral blood lymphocytes. When looking at this as a regimen and looking to see how long we can have this achieved with just a simple selection criteria, the question is would a patient always have to be on a selection criteria in order for this to work long term? The short answer is in our animal models, we were able to do this over a very short period of time. Within essentially 4 weeks of the selection pressure of the chemical entity, we were able to actually drive up the bone marrow and keep up the peripheral blood lymphocytes. So again, the concept here is every patient is different, and depending on the level of expression necessary within the patient, you may need to give patients more or less. Nice part about this is this selection criteria in 2, 3, 5, 7 year time period, if for some reason a patient's own marrow was compromised or comorbidities ended up causing a reduction in the expression, you'd be able to redose or boost the patient. So for many of you, I'm sure you're familiar with sickle cell disease. I'm not going to go into all the specifics, but it's a point mutation in beta globin gene, which essentially results in sickling. Millions are afflicted worldwide. Here in the US, it's about 100,000. And after a patient is about 18 years of age, the context of severe sickle cell disease comes into being. Vaso-occlusive crises, a whole host of different issues with pain and the like. The current standard of care is hydroxyurea. There is a curable path, which is allogeneic transplant, but unfortunately it's only about 10 to 12% of patients that qualify under that. And out of those 10 to 12%, only about 50% of those patients choose to go down that path due to a whole host of restrictions and concerns, everything from the sterility of their children and fertility, but also concerns with graft versus host disease and the amount of time that it actually takes for patients to recover. So stem cell gene therapy has the ability to address sickle cell disease. There's a number of groups that I'm sure you're familiar with, including Bluebird, that have really shown that this is a path that can be essentially rendering patients asymptomatic. We're excited about that. We believe that with about 30% expression of anti-sickling in these patients, it's been shown that you'd be able to render these patients asymptomatic. And so the essential piece here, though, is that patient-to-patient variability, unfortunately, may make it difficult to achieve that, hence the selection technology. So what we are doing is we are coupling our backbone, our Cytegrity, which is a stable production system that allows us to produce at scale, with the selection technology, and we're coupling that with a self-inactivating lentiviral gamma globin construct. It's been modified to include a mutation that basically improves the affinity for alpha globin, and we are able to distinguish this once the patients are able to have this within their system, and we actually will be able to identify HbF levels specifically, which is the fetal hemoglobin, which is the anti-sickling within the system, within a short period of time. Within the mouse models that this has been done, the Burke sickle model, you can see that we were able to correct organ pathology, essentially between 50 and 60% reduction in spleen weight. And then what's been remarkable in the animal studies is that there have been no liver infarctions and no kidney pathology was detected. An important study, of course, but animals are not humans. What's even more important, and was certainly a process of learning about just the impact of some of these aspects pathologically, but also on survivability. Once we achieved higher levels than 10% in the mice of fetal hemoglobin, we ended up having a significant impact on survivability. So our next milestones as a company, we are moving quickly to move into the IND. We expect that by Q1 of 2018. And with the ability to do outpatient stem cell gene therapy and the accelerated possibility of being able to increase or titrate up the level of engraftment, we see that by second half of 2018, we'd have early data and proof of concept with this new technology. What's nice is being in the space for a while, we've been able to bring not only a core technology to the mix, but have enhanced the team over the last couple of years, including bringing on board members of the team that have been part of very successful companies, including investors like AbbVie and RA Capital. As it relates to just the last step here as a company, this is a partnering event, recognizing as such. Calimmune has spent a tremendous amount of time identifying technologies throughout the years that we've connected to in-house work. We look forward to being able to work with a number of companies beyond the spec and the work that we do. Lenti and the like is certainly capable and works in CAR-T and other programs, and we're interested in partnering beyond our specific areas of expertise. Thank you.